welcome for this evening's seminar, Judi Smith.
Thank you very much. I appreciate you coming out on a Saturday night. What we do at the International Clinic of Biological Regeneration, ICBR, the only treatment we do is cell therapy, C-E-L-L. Not to be confused with stem cells, which I'll try to explain the difference a little bit later. This is a treatment that really predates that by quite a bit. It is a general age management treatment. There was a Dr. Paul Nahans, which you may have heard the word. There are cosmetics, from Clinic La Prairie, and that is Dr. Nahans, a continuation of his practice. But he began in the 30s and 40s doing research that showed that the injection of animal tissue could rejuvenate our organs. His research was ongoing, but he was a general practicing MD. And so happened he had a case in the hospital, or was called in on a case of a woman who had had a thyroidectomy. And her parathyroids had been inadvertently damaged or removed, which resulted in catastrophic muscle failure. That's what controls your muscle contraction. And so she was dying. Her breathing capacity was diminished. She was going to lose her ability to swallow. It was fatal. So he was called in to see if he had anything to offer, and he, building on his research, did the crudest thing you can imagine. Went to literally to a slaughterhouse, got a young calf, macerated the parathyroid glands, took him back to the hospital, couldn't have been less sterile, and injected them directly, into her pectoral muscles. So be it, in those days, because there's no malpractice, you live or die, and, he did the best he could. Well, she did survive, and she did regain her function. She lived into her 90s. They were friends for life, outlived him, as a matter of fact. And that was the beginning of clinical cell therapy. So with that success behind him, that really wasn't what he had in mind, was that he really looked at it as an anti-aging, age management therapy. And so in the 40s and 50s, he broadened that to that aspect of cell therapy, and he became very, very well known for treating some of the Hollywood stars of the 50s and some of the post-World War II dignitaries and statesmen from World War II. The Pope Pius XII was a patient. That really put him on the map. So he opened Clinic La Prairie, which was the first cell therapy clinic. So basically, the idea is to use tissue of a young embryonic or fetal animal and to inject them. And in those days, it was called live cells because it actually was taken in a semi-surgical procedure from the embryonic animal and injected into the patient within 60 to 90 minutes to preserve the live cells. Well, what really happens after about 60 minutes is they deteriorate too much for injection. So that's why it had to be done so quickly. It was an effective treatment, and he certainly got a reputation for the results he was getting. It also had some danger involved, as you can imagine. Anytime you're injecting whole cells, literally a macerated organ, you're going to have extracellular material, you're going to have foreign protein. So they had to be kept, it's quite a procedure. It's semi-surgical, they had to be kept in-house for 3 to 5 days at least, and that's if things went well. There would be a slight fever. They would, in many cases, develop deep muscle abscess, or in worst case, anaphylactic shock. So there was a downside, but it was so popular because it was so effective that people, flocked to him. Extremely expensive. These were just very, very wealthy people who could afford such a thing. Well, it did expand beyond that. By the time we entered the field, and I'll give you a little background, I've had cell therapy clinics since 1981. We've been doing this for 30 years. We have 3 clinics. They're in Nassau, Bahamas. We have 2 in Mexico, a very small office in Matamoros, Mexico, which is south of Brownsville, Texas. We've had that for years. And then in 2007, we moved out to Tijuana. So that is basically what we do. It is the injection of these embryonic and fetal cells. Now, we've developed a protocol over the years. We filtrated it down to just the genetic portion of the cell. The other parts of the cell are extremely important for the life of the cell. But therapeutically, we're only interested in the genetic code, the RNA, DNA of the cell itself. So now they have it down to that, and it naturally breaks in these smaller sequence the way the RNA goes out and builds new cells. So that has removed all danger from the treatment because there's no longer protein in nature. Nano-sized particles makes for much better absorption. We can get billions and billions of more cells in a reasonable size injection, and it's very, very safe. These are not stem cells. I don't want to mislead or confuse anyone. Stem cells are completely without identity. They're the earliest embryonic cells. And stem cells are rapid growth. This is why stem cell therapy is going to be such a miracle. They're going to be able to use it for things like Parkinson's disease, diabetes. The most exciting, as far as I'm concerned, is spinal cord injuries. You're going to be taking someone paralyzed in an accident, get them to the hospital just like you would a broken bone. They're going to inject the stem cells, heal that site of injury, and have them recover. Really exciting things with stem cells. But they will be used for specific things. The stem cell has no identity. It has to be surgically put where you want it, whether that is a site of injury or a site of disease. If it's a spinal cord injury, it has to be surgically implanted at the area site of injury. It is a specific treatment for a specific condition. Cell therapy is a more general treatment. We're using cells that are of a more mature nature. So if you kind of think back to your 9th grade biology where you understand that all mammalian cells are the same at a point of gestation, and then they start to differentiate. Well, they start to differentiate first by organ, then by species, and then ultimately by individual. So these are cells that are taken at a point that they're called organ-specific. If you put heart cells in a petri dish, they will start to come together and start to pump in a rhythm. They have an affinity for one another. So when we inject these cells that are organ-specific into a large muscle mass, this is an intramuscular treatment, by the way, not intravenous. It's an intramuscular treatment. Injected in the muscle mass, they know by radioisotope tracing that these cells will migrate to their target organ within 60 to 90 minutes. There are fundamental things that affect us as we age, and that is a decline in the connective tissue, a decline, which is what makes us look older, a decline in the immune system and the endocrine systems. And those are the things that, that's Mother Nature's way of taking us out. But ultimately, those systems fail. We become open to infection or serious disease. So what we're trying to do is to keep those systems vital, to stay as young as you can, look as good as you can, as long as you can. It's not a panacea, it is not a miracle, and it's not a mystery. It is simply a good treatment that, taken with exercise and a good diet, it's not an antidote for a bad lifestyle, will help to prolong, youth and vitality. There is one serum that's called Omnigen, and that is an overall cellular therapy, whole embryo treatment. We treat with the entire endocrine system. Those are the things that tend to fail with age, and that is a biofeedback system. So I don't know exactly what anyone knows about cell therapy or what you might like to know about it. If you have any questions, feel free to call and ask them. Yes, ma'am.
Hi. Is the injection something that would be safe for someone who has a compromised immune system?
Oh, absolutely. Oh, it would be advisable for someone with a compromised immune system. Absolutely.
Like, let's say someone who has to take prednisone and their immune system is suppressed.
Right. Now, as long as they're on something that is continuing to suppress it, it's a constant, battle. But we have seen people with very depressed immune systems, and that's something you can measure because those white cells change on a daily basis. There are different rates of cell division in our bodies. Soft tissue, as I mentioned, tends to renew once a year. Our nerves very slowly. They used to say not at all, but now they know that they do. But the white cells, the immune function cells, on a daily basis, you measure your white cells 36 hours after cell therapy and see that there is a better distribution. So it is a good thing.